Genetic drift operates in option A, non-reproductive population. It is incorrect because genetic drift cannot operate in a non-reproductive population as there is no reproduction to create genetic variations or alter the allele frequencies. Option B, slow reproductive population. It is also incorrect because genetic drift is not linked to the rate of reproduction. Option C, small isolated population. Now, genetic drift refers to random changes in allele frequencies within a population over time due to chance events. In small isolated populations, the effects of random events are amplified, leading to significant changes in genetic composition over generations. Hence, it is the correct option. Option D, large isolated population. It is also incorrect because in large populations, genetic drift has a minimal effect because random changes are averaged out across a large number of individuals, thus reducing its overall impact. In this question, you have to choose the option that represents the frequency of heterozygous individual as per Hardy-Weinberg equation. The Hardy-Weinberg equation is p square plus 2pq plus q square equal to 1 or p plus q whole square equal to 1. P represents the frequency of the dominant allele, q represents the frequency of the recessive allele, p square represents the frequency of homozygous dominant individual, q square represents the frequency of homozygous recessive individual, and 2pq represent the frequency of heterozygous individual. Hence, the correct answer is option D. In this question, you have to choose the option that represents the correct chronological order of human evolution. Now, Ramapithecus are considered one of the earliest ancestors of humans, followed by Australopithecus, which were bipedal and primarily herbivorous. Then Homo habilis, that were known as the handyman due to the evidence of tool usage, and Homo erectus, which were the first hominid to use fire and spread out of Africa. So the sequence of human evolution is Ramapithecus, then Australopithecus, then Homo habilis and Homo erectus. Hence the correct answer is option D. In this question, you have to choose the option that represents the correct sequence of events in the origin of life. Now first, the synthesis of organic monomers took place. That is, simple organic molecules such as amino acid, nucleotides were formed from inorganic precursors through the processes like those simulated in the Urey-Miller experiment. Then synthesis of organic polymers took place. That is, monomers combined to form polymers, to form protein, RNA, on the surfaces like clay which act as catalyst. Then formation of protobionts took place. That is, primitive cell-like structure are formed consisting of polymers enclosed within a lipid membrane and showing simple metabolism and replication-like processes. And then the formation of DNA-based genetic system took over. That is, the evolution of a stable genetic system based on DNA that allowed accurate replication and inheritance, marking the emergence of life. So the correct sequence of events in the origin of life is 2, then 3, then 1, and then 4. Hence the correct answer is option A.